Welcome to this third and final video on membrane structure and function. In the first video we looked at membrane structure, so this picture should be pretty familiar to you. We've got a phospholipid bilayer with proteins either extending through the entire membrane or just on the surface. These surface membranes are integral or transmembrane proteins as they're sometimes called. These things here, cholesterol molecules, that should all be familiar to you out of the first video. In the second video we considered diffusion and osmosis. And in this one we're going to take a closer look at the role of these proteins, particularly the transmembrane proteins, and consider how water molecules get through a membrane and how other molecules and substances actually go into cells or out of cells. And that'll wrap it up on here. If you need to go back and have a look at one of the other videos in this playlist, I'll put the link to the playlist up here, and of course my gecko for subscribing will be down here, but that's at the end of this video. So let's take a close look at this. These proteins, integral proteins or transmembrane proteins, is what we're going to spend a lot of time on in this video. I want to mention these ones here, these receptors. I'll just on the surface of some cells. These are the things that would take, say, a neurotransmitter that's released from one nerve cell. It would bond into a receptor site like this on a next nerve cell, and it would have some impact on that. So sodium would get into the neuron and things like that. So they bond onto molecules in the surfaces. Another example is protein hormones like insulin. Insulin bonds into a receptor and it has some impact on the cell. Other hormones like steroid hormones, they're lipid soluble. And lipid soluble molecules, they can actually sneak through. They can sneak through these phospholipid heads beside the proteins. And this is a lipidy nature inside here. It's like a light oil inside. And a steroid hormone, for example, like testosterone or estrogen, can actually cross the whole membrane and get inside the cell and play its role inside the cell. Having said that, brings me to the topic of water. We had water as a topic a long time ago, and water is a small polar molecule. Water is in the extracellular fluid. It's in the cytoplasm. It mixes freely on the surface of the cell, both outside and inside the cell. But water can't sneak through. So we talked about osmosis, where water crosses a membrane to equalize concentrations of substances on both sides. Well, it does cross the membrane, but it doesn't cross the membrane by going through there. Water actually goes through proteins. So if we take a look at, I'll just put an arrow through here. A water molecule is actually quite small in relation to this thing. It actually sneaks through the protein. How? Well, if you remember protein structure, this is, say, a tertiary structure of a protein, and it's this type of a thing here. This is the amino acid sequence here. There's spaces in here. And water can actually sneak in between these spaces and get through. These spaces would have to be lined with polar R groups, so polar matches polar and can actually go through. That's how it gets through. These things are called channel proteins. And we talk about them being protein channel. So it goes through a protein channel, a channel through a protein in a channel protein. So it's kind of confusing vocabulary. But channel proteins is the name of them, and they go through a protein channel. So water sneaks through membranes that way. And we talked about how water bounces off the surface to create osmotic pressure. Well, it doesn't happen to slip through automatically all the time. Sometimes it gets bounced off and that's osmotic pressure again. So that's one of the roles of these kinds of proteins, these transmembrane proteins. Another one is, and I'm just going to draw another protein over here, like that protein. If we take a protein that's in a membrane, here's the membrane itself. I won't draw all the phospholipids, it'll take too long. This protein, if I cut it in half now, this protein does have a space through it. Same sort of thing as this, but this one has a particular space through it like that. And what can happen is there'll be a substance in the environment. Let's say it's going to go from the extracellular fluid to the cytoplasm. It's going to go through. This substance can interact, chemically interact with this protein, and when it chemically interacts with it, that's like a before picture, and this can be an after picture, or partially after picture. When it chemically interacts, it changes the shape of that protein. And you see what's happening here. This protein is sort of opening up a little bit, and it takes it in, and that interaction allows it to interact further down. And if I draw a third picture down here, you can see that that substance is moving through, and it will get all the way through. And it moves just because of the interaction of the molecules. This type of protein-assisted transport, and it's protein-assisted. This is a sort of a protein-assisted transport here, but this particular one is called facilitated diffusion. Because it works the same as diffusion. This particle is moving from an area where it's more highly concentrated out here. There's lots of them. Otherwise, it would never have any tendency to go that way at all. But it's being helped in its diffusion process. That's facilitated diffusion. One of the kinds of things that gets through cells this way is glucose. Cells need glucose. They don't have to want to spend energy to get the glucose. This is passive. It's a passive process. No energy required. It just moves by diffusion according to the laws of diffusion because it's more concentrated out here, but it involves this interaction with the protein. Glucose, for example, is too big a molecule to just bounce around and sneak through here, and it probably wouldn't be very successful going from a polar area to a non-polar area to a polar area again, so it doesn't work that way. There are other examples of protein-assisted transport that do require energy. This is facilitated diffusion, does not require energy. The type that does is called, and I'll just erase the word, I could almost use the diagrams again, it's called active transport. And it uses ATP. It requires energy. So for example, it may be that there's lots of these substances inside the cell already, and there's fewer of them outside. It goes against the laws of diffusion, so it takes energy to disobey the law of diffusion. So it's called active, because it's actively using energy to transport substances through a protein in a membrane. Example of this, let me get rid of this here. Example, sodium-potassium pumps. The word pump, already you can see that, oh yeah, but something's being pumped, it uses energy. Your nerve cells actually physically move sodium and potassium across the membrane using these kinds of proteins to restore the resting potential. And that's a topic that's way ahead in the nervous system, but that's what goes on. The sodium and potassium ions get displaced across the membrane, and they have to be put back in their correct position. So sodium-potassium pumps are one example of where that happens. Another example is in the manufacturing or the making of thyroxine. Thyroxine is a hormone. Thyroxine comes from the thyroid gland in the neck. In order to make thyroxine, the thyroid gland has to accumulate iodine. So it takes iodine out of your blood, and the cells of the thyroid gland actually use iodine. The concentration of iodine in those cells is about 25 times as concentrated as it is in the plasma itself. So it's actually accumulated. It's moving against the laws of diffusion. It accumulates iodine in order to do that. So there's a specific examples of this, and I'm going to just show you. I'm just going to make sure you understand that some of this is what we call gated ion transport, where you have gated ion channels, I guess is a better way to say it, because we do have these channels, we do have these chambers that go through here, and whether it requires ATP or not, sometimes they are operated as gates, and that's why we use the term gated ion channels. So these things could be ions, as in the case of iodine here, sodium and potassium there. They don't have to be ions, but gated ion channels could be operated by the presence of a ligand. Now a ligand is just a molecule. It's a fancy name for a molecule. So what happens is a molecule, I'll use a different color here. A ligand, let's just say this is a ligand, will attach to the end of the protein, and by attaching to the end of the protein, it actually encourages it to change its shape, and it'll open it up. So a ligand operated ones, we can have those which are mechanically operated. So these channels will open up by pressure mechanically, or even that, if you take your hand and you chop your arm, you're going to get a nerve impulse generated. Well, it actually goes in both directions, but you're going to feel it. The nerve impulse is generated because you've mechanically stimulated the nerve cell and it's opened the channels. So the ions will move. And the third way is actually by voltage. So these gated ion channels can be operated by three different systems. By the presence of ligands, or mechanically, or by voltage. And a good example of the voltage one is back to the sodium-potassium pumps, because in the nerve cells, there's a potential difference, there's a voltage difference across the surface of the nerve cells. So, with that, I hope we've got a better understanding of what these transmembrane proteins are like. So we get things that are crossing membranes according to the laws of diffusion, and some which are crossing not according to the laws of diffusion. Cells even get bigger things to cross their membranes, bigger than molecules. These ones also require ATP. So if we have to make a dividing line, when we had facilitated diffusion here, facilitated diffusion, osmosis, and just regular diffusion, none of those require energy. This one requires energy, and so do the next two. I'll leave my picture of the membrane there. The final two are what we call endocytosis and exocytosis. So just as a quick description, endocytosis, endo being into or in, exo is out, cyte is cell. So this is where things are moved into a cell, and this is where things are moved out of a cell. But not just moved, like these things move like that, but this is through the use of vesicles. Things are put in vesicles, and vesicles are membrane-bound chambers in cells. Cells make vesicles, they make them and release things by exocytosis. They make vesicles on their surface and take things in by endocytosis. So I'm going to show you what that looks like. But before I do that, this topic only works for eukaryotic cells. Prokaryotic cells do not have membrane-bound organelles of any sort, and that includes vesicles. So this process, or this type of process, only works with eukaryotic cells. So let's look at endocytosis. If this is, I'm not going to draw the membrane that well, I'm just going to draw it like a single line. We take that membrane, shrink it down like that, and if there are substances in the fluid, extracellular fluid outside that the cell recognizes chemically, so there's a chemical interaction here, what can happen, I'm going to draw a series of diagrams, this can give way to this. It'll actually have a slight invagination here. The term invagination means it punches in or pushes in a little bit. So these things collect in this little dent, and if the process continues, the little dent closes off like that, and you get a little piece of the membrane. That's membrane. This is the membrane here, this membrane-bound vesicle. And these things are inside. So that could be chemicals in the environment. We call this process cell drinking. You know, in general terms, because this endocytosis process, technically the cell drinking is pinocytosis. What happens is these chemicals which are in the fluid around it get taken in, but so does the fluid. So it's really, we're talking about small particles, and it's a type of, that's why we call it cell drinking, but pinocytosis. What happens to the vesicle? The vesicle goes to a lysosome. Now, lysosomes are part of the endomembranous system in eukaryotic cells. Lysosomes contain hydrolytic enzymes, and what happens is it'll fuse with the lysosome, it'll take apart anything that's in here chemically, it just breaks it down into unit molecules, if you like, and then it can use the molecules, and if it's just water, it uses the water in the cytoplasm of the cell. So that's pinocytosis, or cell drinking. But what happens if the cell recognizes something that's bigger than little particles and bigger than water molecules, and it wants to take those things in? Well, that's still an endocytosis process, but it's not cell drinking anymore. I can erase this. I'll draw something a little bit bigger. It's going to look a little bit different than this. Then I'll draw the same sequence though. It's not called cell drinking. It's called cell eating. So that implies right away that it's bigger substances. And it's called phagocytosis. Instead of pinocytosis, we have phagocytosis. And a really good example of this is amoebas. An amoeba has been seen actually literally eating things. It sends out what are called pseudopods and engulfs it. So here's what that process looks like. It may be that this is the surface of the cell again. Here's a substance, it may be a bacteria cell. Quite large then. And there's chemicals on the surface of the bacteria. There's chemicals here, receptors, and all these kinds of identity things. It'll recognize, hey, wait a minute, that thing's food. It wants it, it'll take it in. So what happens quite simply is it will, there's the bacteria, the cell. It will actually take its membrane and it'll do this. It'll go out and grab it like that. And it'll go out and extend some pieces of the membrane, these things here are called pseudopods. False feet. Now the reason it has that name is in a large sense, if you look at the amoeba, an amoeba is one of these cells that has random changeable shapes. These things are called pseudopods, and when amoebas encounter food, they literally send their pseudopods out and engulf it like that. So even in cells which aren't as active as amoebas, they still carry out phagocytosis. And if we draw the next picture, these pieces will continue to grow around, and it will grow closed. They will fuse together, and again we're left with a vesicle, and the vesicle has the food substance, bacteria cell or whatever, inside it. This, again, goes to the lysosome, because the lysosome is going to digest the contents, and then the cell can use those contents. There's one other variation of this, and I'm going to use the same diagram again, and you can just see the increase in complexity here. This one doesn't have a name like that. It's going to wind up looking very similar to this. It's called protein receptor-mediated. It's a protein thing, but a receptor-mediated phagocytosis or endocytosis. Better call it endocytosis. In this case, what happens is there's specific receptors on here, and there's an interaction between the receptors of the cell membrane and the surface proteins of whatever the substance is out there. And if I look at the next picture now, the next version of this, these things actually interact, and you can see they're being held in here, very similar to what the other picture was. This will grow closed, and you get that sort of thing there. The difference is that these receptors, there's a protein called clathrin. Clathrin will actually coat this thing. Once it does this, clathrin is put out here, so you get this clathrin-coated vesicle. So it's got an extra protein layer on it. Clathrin, and it gets digested by the lysosome as well. So that sort of summarizes this whole thing. We have looked at the ways things cross membranes, from whether it's as simple as water molecules and ions sneaking through proteins, or facilitated transport, active transport, and now we've looked at actually 3 different versions of endocytosis. Exocytosis, really exocytosis is a topic coming up because it's part of the endomembranous network in a cell, but what exocytosis is almost the reverse of that, where a cell will produce a vesicle and it's got contents in it. It will go to the membrane, it'll fuse with the membrane, and the contents can be expelled out of the cell. That's exocytosis. Cells release things that way. So if we're talking about cells which make insulin, I've talked about insulin already, cells which make insulin, the insulin gets put in vesicles when it's being made, and it gets released into the circulatory system by all the beta cells of the Langerhans in the pancreas. So I think that wraps it up here. Again, if you want to go back and look at one of the other videos, the entire playlist, put it up here, the gecko down here, and thank you very much again.